So it's a pleasure to introduce the next speaker, which is Bram Heijs from Leiden University Medical Center. And he will be talking about spatial dynamic metabolomics.
Yeah, thank you for the introduction, Guinevere. Yeah, slightly misleading title here. Made a mistake. I hope that will be the only one for today. So indeed, mass spectrometry imaging, the title is true though. We can use mass spectrometry imaging as a tool for multi-omics in the spatial nature. But today I want to talk about dynamic metabolism. So we're adding a different dimension to 2D data, which is time. So brief introduction, maybe a bit redundant. Should have looked at the program better. We apply direct molecular imaging using mass spectrometry imaging. So we take a fresh frozen or formalin fixed tissue section. We apply sample preparation that defines basically the omics layer that we look at. And the sample preparation always includes, since we're using MALDI mass spectrometry, MALDI matrix application. So then we apply this spatially correlated mass spectrometry at a user-defined spatial width for single cell resolution. This is typically 5 by 5 micrometer. And then we get these cell type specific mass spectra. So we can recreate molecular images at the single cell level. And since this technology is relatively non-destructive to the tissue, we can supply the tissues that we have analyzed by mass spectrometry imaging to post-MSI analytics, such as immunofluorescence staining or histological staining. As you've seen before, we can extract molecules and identify them by LC-MS. And of course, we can do all the univariate and multivariate data analysis strategies that you have seen before. So basically, today's talk will be mostly about in situ metabolomics using mass spectrometry imaging. And here on the bottom, you see a typical mass spectrum, average mass spectrum that you get. If we look at a kidney section by mass spectrometry imaging, so we've used NEDC as a MALDI matrix, and we analyze in negative mode. And basically, on the lower end of your mass charge scale, we see small peaks that represent small molecules, such as lactate, hexoses, malate, whatever. All the glycolysis and TCA cycle intermediates. Of course, we also get a lot of interference from the MALDI matrix. That should be a disclaimer there. And in the higher mass range, we see our glycerophospholipids. And as you can see here from the coronal section of the kidney, they exhibit they have a lot of cell type specificity. So we have these structures here, for example, that represent the S3 segment of the proximal tubule, and then we have some features that highlight the glomeruli and other parts of the nephrons in the kidney. So keep this in mind, this will come back later. So what we should realize, and I think that's mostly the case of all the talks on spatial omics that we have seen today, is that all these images that we look at, they reflect snapshots of highly dynamic processes. And what we want to show, what we set out to do actually is create a method that sort of reflects the dynamic nature of these dynamic processes that we look at. And one of the ways to do this in metabolomics is by using isotope tracing, and I don't claim that we developed it. This is a complete field on its own, and they call it fluxomics. We cannot call it fluxomics since we don't actually calculate fluxes, so we refer to it as dynamic metabolomics. So what it's based on is the introduction of a labeled nutrient into an active metabolic system, and basically what you look at is the dilution of the isotope labels, in this case 13 carbon, throughout the metabolic pathways into the endogenous compounds that are produced after introducing the flux compounds. And basically the advantage of mass spectrometry in this case is that these compounds, they obviously differ in mass, so what you will see over time is a decrease in the endogenous 12C compound and an increase in the 13C compound and the labeled compounds. And since they differ in mass, we can very nicely detect this by mass spectrometry. Since we can detect it by mass spectrometry, that also means that we can detect it by mass spectrometry imaging. So that's what we again set out to do. So we have developed this complete workflow. I will walk you through it, don't worry. It's a complex figure, so I will break it into smaller parts. So we start by harvesting a tissue from, in this case, a mouse. So we harvested a fresh kidney and we subjected that to vibratome slicing. We create 350 micrometer thick vibratome slices, which we then incubate in a defined culture medium where we start to introduce at specific time points 13C labeled 13C labeled nutrients. So we did that with glucose, linoleate, and also glutamine. And as you can see, all these different compounds, they start to produce different intermediates of the metabolic intermediates downstream in endogenous metabolism. So we then took those samples, we flash froze them using liquid nitrogen, sectioned them, went through the entire procedure for mass spectrometry imaging, did post-MSI immunofluorescence to basically validate our cell types afterwards, and end up with a big pile of data which needed to be integrated. And to integrate all this data, we sort of took a similar approach as Jasmine showed us earlier today. Basically, she showed this workflow where you take a single-cell RNA sequencing data with a lot of genetic coverage, so a lot of transcriptomic coverage, and you take a spatial transcriptomics data set where you have spatial information for a limited subset of those genes. Those 1,000 genes will very likely also be present in the single-cell RNA sequencing data set, so basically when you do your segmentation based on those 1,000 genes, you can basically create profiles for different cell types, for different cell types, and imply the information of the 24,000 other genes onto the spatial coordinate system that you have from the spatial transcriptomics dataset. And by that you get unmeasured gene prediction. And basically if you predict genes that are actually present in the dataset, sort of a cross-validation, you see that the measured and the actual predicted intensity patterns and localization patterns look fairly similar. So we took this approach and applied it to the mass spectrometry imaging data sets that we have, basically changing here the information content from transcriptomes to lipids and metabolites. So basically what I told you before is that these lipid profiles are very cell type specific, and that means that we can use them as common information to link these different data sets together. But there's one caveat here. We introduced stable isotope labels, so we should confirm that these stable isotope labels do not affect the lipid profiles that we actually detect. So that's what we did. We had a look at how these lipid profiles change, or whether they change, and we found that they pretty much don't. We still get the same cell type specificity for the same set of analytes that were added. And also then, if we do the imputation of the metabolites between the different data sets, we see that indeed the cross-validation of imputation versus detection is actually pretty good. So we were confident that we could do these measurements on the samples that we wanted to analyze. So all this work, as you can probably see from the samples that we have used, has been done in collaboration with the Department of Nephrology. And what they actually do is, forgot one slide, so they have, not a mistake. So there we have our combined data set, and we can calculate per pixel metabolic rates, or we can actually look at the dynamic metabolics and create images in which we reconstruct the metabolic activity of the tissues that we have analyzed. So again, we did this in collaboration with the Department of Nephrology, and there they are really looking forward, or looking to create kidney organoids and use them in the future as a way of solving the shortage of donor kidneys by being able to transplant kidney organoids into a human setting. However, one of the problems that they currently face with these kidney organoids is that they actually don't. If you actually look at the development of the kidney, you have these proximal tubules here, and these proximal tubules, they are not really formed well in the kidney organoids in a dish. So what we did is we took human fetal kidney, subjected it to spatial dynamic metabolomics as well as single-cell transcriptomics, which gave us a single-cell transcriptomics data set, which is fantastically suited to pick out all the different cell types you get throughout the entire development of the human kidney. We then actually chose to only look at metabolic genes, and we found that actually these metabolic genes were very similar to the complete transcriptome, are able to define the different cell types that you find in development. And actually, if you look then at the pseudo-time analysis, it's not only able to identify different cell types. It's also able to reconstruct the developmental pathways that you say, that you would find in a tissue. Which means that actually the metabolism is very important in making cell fate decisions during embryonic development. When we then applied our metabolomic imaging, so basically the clustering you see here is on the basis of all the phospholipids we detect, we can create these spatial segmentation maps, and you can really nicely see that all the different segments of the kidney really match the different cell types that we can confirm by doing immunofluorescence staining. And then if we also here look at the trajectory analysis, we also see that these lipid profiles are very capable of following the development of these tissue sections over basically tracing the lipidomic profiles. We can then reconstruct this into an image again, and what we find indeed is that we can really nicely trace the developmental trajectory based on lipid profiles. So all this lipid information comes from the same analysis, from the exact same analysis that we recovered that we did to recover the spatial metabolic information. And that's what you see here. So basically we now take all the cells that are from the renal vesicle stage, the S-shaped body stage, that are developed further into the mature proximal tubules. And if we look at the single-cell RNA sequencing data, we learn that in this development pathway, we see a reduction of glycolysis, an increase of the TCA cycle, and an increase of mitochondrial fatty acid oxidation. So what we then learned based on tracing the glycolysis using uniformly labeled glucose as a nutrient for these tissue sections, as well as linoleate, we were actually able to confirm that indeed this pathway indeed draws most of its energy nutrient consumption from linoleate rather than glucose, and it changes over time. So indeed, the development of proximal tubules is associated with the metabolic switch from glycolysis to fatty acid oxidation. So now that we knew this, we wanted to implement this in the organoid culture that we were actually interested in from the beginning. So when we compare the fetal kidney lipidome with the lipidome from organoids of two different iPSC cell lines, we see indeed that the cluster with the lipid cluster with the proximal tubules is basically missing, with mature proximal tubules, is basically missing in the organoid culture. So we decided to feed these cells with regular linoleate, and that really didn't work into creating any mature proximal tubules. We then found out why, because one of the long-chain fatty acid transporters essential for getting the fatty acids into the cell is missing. We actually then found that there is a short-chain fatty acid transporter available in these cells, so we started to include, rather than long-chain fatty acids, short-chain fatty acids such as acetate and butyrate. And lo and behold, once we started to incubate these organoids with short-chain fatty acids, we saw the production of mature proximal tubule species, which were then confirmed both by staining and by the molecular profiles that we detected in these cells. So as a conclusion to this part, I would like to, I hope I've been able to convince you that mass spec imaging can be used for spatial multi-omics. But moreover, that the combination of mass spec imaging and isotope tracing is a very powerful tool for analyzing metabolic dynamics, and that we have all kinds of different biological findings we can derive from this. So then there was also sort of the assignment from the organizing committee to sort of give your idea or vision where single-cell multi-omics or multi-omics in general is lacking. And I think if we want to go through single-cell, we should start from, we should consider spatial information. So we have now already been able, and we've seen some examples today, that we can combine a lot of different imaging modalities on consecutive sections from the same sample, or even from the same sample, or even from exactly the same sample. And one of the important things to consider here is not only the computational integration, but actually also the compatibility of all the different analytical approaches that we would like to look at. And then I think where the biggest challenge now lies is not only this integration, the successful integration of all the modalities, but actually adding depth to context. And I think Casper gave a very nice presentation before, showing that we indeed should consider these microdissection approaches and supply them to, subject them to very sensitive glycomics, lipidomics, and glycoproteomics approaches to indeed add the relevant context, relevant depth to the right context in which cells persevere. So with that, I would like to thank a lot of people that were involved, specifically Gangqi Wang, a very talented postdoc who unfortunately wasn't able to give the talk today. And I would like to thank you for your attention, and I'm happy to take any questions.